Okay, we're going to start discussing proteins in a more specific way with the receptor proteins. I know that I promised last time I was going to start with cell adhesion molecules, but I changed my mind. And that's because the receptor proteins are kind of where it all starts. And I thought it would be a logical way to begin. So, less about me, more about the proteins. Receptors help cells do very specific things in very specific situations. For instance, jump cut, kidding. What tells you what's going on in the world around you? Information comes from your ears, your eyes, your cell phone, and then you can decide what to do next. But cells have to make those same decisions without, ears or anything like that. So, how does a cell check its Twitter? That was a lame joke. Anyway, how does a cell figure out what's going on around it so it can decide what to do inside it? And the answer is chemicals. I don't mean chemicals in the sense of pesticides and food coloring. In your body, pretty much everything is a chemical. Get used to the word because it's a good one. So we're going to be talking about receptors and ligands. Ligands. They are molecules. And we'll start with receptors. Basically, a receptor is a protein that's in charge of catching things outside the cell and responding to them, doing something because of them. A ligand is basically a word that we use for something that binds to a receptor. So you've got a receptor, anything that sticks to it is a ligand. It can come in all different shapes, it can be all different things. Some examples of ligands are neurotransmitters, things like serotonin in the brain. Smells, the things you smell are actually little molecules that actually physically get in your nose. So when you smell poop, draw your own conclusions. Hormones in your body make different cells in different parts of your body respond to do different things, all sorts of different things. And a great example of a ligand is, well, there are so many of them I ran out of space. So an illustration of a ligand receptor relationship would be this, where the gray squiggly fancy thing is an example of a receptor. And you see how it kind of crosses the cell membrane 3 times, down, up, down again in a little loop. It's very common for receptors to cross the cell membrane up to 7 times, and there's a whole family of them that does it specifically 7 times. So you've got the part of the receptor that's outside the cell, and that's got, it looks like kind of an open catcher's mitt, an open hand. And then you've got the part that crosses the cell, and then inside the cell is the part that does something neat. So the little blue paisley guy coming towards it would be the ligand, and the idea is that you've got a cell with stuff sticking out of the cell membrane, such as this receptor, and it's got goo floating around the cell. We haven't really gotten into the goo much, but there's goo around cells, and blood, and lymph tissue or lymph fluid, and all sorts of other things. And in that goo floats ligands. And so this ligand is shaped just right so that it binds to that receptor. And when it does, the neat thing is when the receptor connects to that ligand and feels the bind, it actually changes its shape and alters what it looks like inside the cell. So it changes its shape inside the cell and opens up, in this case, in my cartoon case, it opens up and lets the little green circle free. So see, it goes, vroom. Yeah. And then the little green circle goes off on its happy path to go do more things in the cell, and we'll learn about that soon. It's really cool. So The first step is the right shape, fits into the right spot on the outside of the receptor, outside the cell. Step 2, the receptor changes its shape inside the cell, which is just what we saw in my crazy, can we call it an animation? I'm calling it an animation. And 3, a party starts inside the cell. This is called signal transduction. And signal transduction is a fancy word with an easy definition. Think of it like a waterfall or something falling down a staircase. Step by step, it's triggered by something sticking to a receptor, and then for instance, in a picture, the receptor let go of a little green ball, and that rolled down and stuck to maybe something else, or bumped into something and made it move. All sorts of different examples that we can think of for that. And it's like a Rube Goldberg machine, if you've heard of those, where the marble rolls down and it brushes your teeth for you at the end. I don't know. So I should have drawn one of those. Ooh, you might never see this video. I might redo it. Here's another drawing representation of a receptor. On the left, it's the part outside the cell. It's only crossing the cell membrane once because I'm lazy. And on the right-hand side is the intercellular part of the receptor. So say the pink thing is its ligand. It comes along, and when it binds or attaches or docks like a spaceship, it changes the shape, opens up the inside, and lets something else go into the cell. So another high-tech animation. And this is my last illustration of a more complicated receptor issue or situation. And it shows a little bit more of the picture. First of all, the receptor has 2 binding sites. They're both bound to pieces of the same ligand, although that's not always necessarily the case. Sometimes you need multiple ligands to be present at the same time to cause a certain effect. But see how that green hexagon on the left is a ligand? That hexagon is a shape that you find a lot in things that have smells. We may or may not get into that. It's just a chemical shape that really is a hexagon with atoms at each point, and they're bound together along the edges. And they are very aromatic. They're called aromatic hydrocarbons. And so this is the kind of receptor you might see in the nose or in the nasal pathway to the brain. So the idea is this cell would have a receptor on it for the smell of cheese. And that's a little cheese molecule, cheesy smell, fragrance molecule. And it binds to the receptor. And inside, the receptor is, now that it's bound, it's changed its shape, it's letting go its own bunch of ligands, which are attaching to receptors inside the cell, which are actually not called receptors, they're called enzymes. And it's very common to find enzymes like that hanging around just inside the plasma wall, just inside that cell wall, so that it can grab onto stuff that the receptors are spitting out. Very exciting. That's it for now.